I'll give a very brief introduction. Leon, I believe, got a BS in chemistry at Cornell, went to Iowa State for both his master's and PhD in entomology and related fields. Leon does a mix of conservation biology, entomology, and forensic science, and I think the latter is going to play a big role in what he's talking about today. And as someone that spent a lot of time looking at dead animals in the field and trying to figure out what killed them, I'm super excited about today's talk, so hope you are too. Thanks.
Cool. Thanks for coming this afternoon. You guys can hear me okay with this mic? Great. Well, what I wanted to do was share with you some of the research that we've done over the last 25 years. And I thought I would do it in the context of talking about a homicide I investigated right at the start of my involvement in forensic science in 2003. So that's sort of the game plan. I also will give a little bit of a background about decomposition before we get started. And so as I figure out how to go to the next slide. So animal bodies, I guess it's also true of plants, but represent sort of an island, if you will, of resources. And they're very transient islands of resources in that lots of things want to exploit them. Unlike a conventional island where we see succession with vegetation and formation of soil and those sorts of things, we kind of see the reverse when it comes to carrion, where we see the gradual dissipation of the resource. And so there's a lot of selection pressure for the organisms that use it to get there quickly and to exploit the resource before other things can. Principally, that's going to be three things. It's going to be insects, microbes, of course, and then vertebrates. And we'll talk about each of those sort of as we go along. Now, the insects, from a forensic point of view, provide kind of a yin and yang sort of thing. On the one hand, the insects are in the process of destroying evidence. So from that standpoint, they're bad for forensic science. They're literally consuming a victim. On the positive side, they provide all sorts of potential evidence. Some of the non-entomological evidence that they can provide are things like DNA samples and drug analysis and drugs. Turns out that the guts of insects don't process very quickly, so that most of the material in a larval gut stays that way, stays undigested for a long period of time. So you can actually recover victim DNA out of insect guts, and you can certainly recover evidence of drug use or other toxicants out of there. So sometimes they'll point to a suspect. I have a lot to say about that. The three big things are location of death, manner of death, and the really big one, and the one in which I'm most involved, is determining time of death. Location of death, there's lots of different ways that can go. One easy way is just the species of blowfly. Blowflies are the number one insect that's going to come to dead animals and dead humans, and they differ in their association with rural areas and urban areas. There's other ways you can use insects. There's a famous example of a guy in Indiana who was suspected of killing his girlfriend and dumping her body in Arkansas, and he claimed he wasn't in Arkansas, but there were dead butterflies that were unique to Arkansas on the bumper of his truck, on the grill of his truck. So there's other ways that insects get used. Manner of death is a test I'll put you guys to. Ordinarily, insects lay their eggs only at openings in the body. And if you do a quick mental count, there should be either 9 or 10 of these. I'm pausing to let you try to do that count and see if you get it. The presence of insects someplace other one of the 9 holes or 10 holes is an indication of a potential wound site. So this pig is not one from my own studies. This is from my colleague Neil Haskell. And I'm pretty sure it's been shot rather than had a humane use of a humane killer. But you can see that there's a cluster of flies here up behind the ear. They're around the wound site, and they'll lay eggs there. Part of the reason that blowflies and other flies have this behavior is that their immatures have really crap mouth parts. They're just a pair of little hooks. And so they will engage in extraoral digestion and secrete a lot of digestive enzymes and then slurp those up. Turns out that's also a reason why they like to feed in communities. And as we'll see, that's problematic, both for forensics, but also ecologically problematic. The picture I showed you is one of a series of Chinese, and the picture I showed at the start of the seminar of the dead courtesan is part of a series of art that's used to depict decomposition. It comes from ancient China and Japan. And by ancient, I mean around 700 and 800. And then the tradition carried forward. They talk about the 9 stages of decomposition. French writing early on decomposition in the 1890s talked about 15. I put up here 5, but to be honest with you, I think there are only 4 stages. And the reality is it's a continuum. And so where you're dividing that continuum is kind of arbitrary. So we've got, as we'll see, the fresh stage, which is just right after death. It's worth noting that you might think that there's not a lot going on in the minutes after death. Actually, there's catalysis and sort of the metabolic systems of the body running amok as they break down. But there's also something going on with the immune system. And I say that because flies have been recorded to go into a dead body, in this case, a dead pig, as rapidly as 5 minutes after death, which I find almost unbelievable. I have seen myself flies trying to lay eggs on dead pigs at 15 minutes after death, which is still pretty fast. So I've joked with students for years about this. There's only 2 explanations. There's the scientific explanation is that shortly after we die, decompositional microbes take over and they start producing different organic volatiles, like compounds like cadaverine and other high sulfur containing compounds that apparently the flies hone in on. The alternative hypothesis is that the flies detect the departure of the soul. That's the problematic because the Catholic Church claims that animals don't have souls, and yet we see it with pigs. So I'm not qualified to explore the theological side of it, but I can say on the scientific side, there's lots of evidence of high sensitivity of flies to these chemicals. And they're robust flyers. As long as the temperature is warm enough for flight, they will find a way to get to the body. And for those of you attending or watching in an effort to try, it's sort of a how-to do murder, which I guess I don't recommend, but who am I to judge what your reasons are for attending a seminar like this? It's really problematic to hide a body from insects. My advice would be to put it in a freezer, but then you got a dead body in a freezer. It's not the best of ideas. They will easily get into a sealed car, into a trunk with minimal delay. This I think was one of the first or second cases I did. One of the third or fourth was in a brand new apartment building not far from just a quarter mile of where I live. And the body was put inside a closet, wrapped in blankets. It presumably was nicely sealed and air conditioned. That didn't stop flies from getting to it and infesting the body. It's bodies in trash bags with twist ties. That too is not enough. The flies are very persistent in trying to get to fresh material. What's that? Um, 3 meters, 3 meters, you're pretty good. Flies will go down, and that's a lot of work, by the way, to dig a 3-meter hole. But yeah, flies will go down into the soil, and there's specific fly species that are known for, they're called coffin flies, that are specific to getting into the soil and getting access to bodies there. It's actually, the whole soil thing is a problem. I'll show you here in just a second, but one of the things about, presumably if you have a body, you want to get rid of it. One of the keys to getting rid of a body is high oxygen content. The more oxygen content there is, the faster the microbes will decompose the body. And so one of the things the insects and vertebrates especially do is they open the body, form tunnels and openings so that there's much more rapid potential for decompositional decay. Eventually, the esophagus will close and you'll go to the bloat stage. And the bloat stage immediately follows the fresh. And what is immediately? I don't know. A day and a half, 2 days, depending on temperatures. That's where the abdominal cavity fills with gases. And years ago, when I was teaching forensic entomology, I had to dig pig out for my students behind the vet school. And I took them out and we had the pig in bloat stage. And one of the students asked me if the pigs pop when they get in the bloat stage. And before I could say anything, the same student kicked the pig. And so I guess this is part of the active learning that I've heard about, because it popped and decompositional fluid went everywhere and students scattered and I laughed. So that's the bloat stage. Active decay is when you have a large group of insects that are consuming a significant amount of tissue. And so this is a classic, what's called a classic maggot mass. You'll see it's completely skeletonized the head of the pig and is now moving into the torso. That confirms the statement that rot begins at the head. Yeah, that's largely true for decomposition. Other things to the side, usually you will see decomposition begin that way. The other thing you should know is that this black circle in the soil, that's from the, it's called purge fluid or decompositional fluid, or stinker juice is what one of my colleagues calls it. Those are decompositional fluids that have poured, that's the result of decompositional fluids that have poured into the soil. And the very high nitrogen content has killed the grass. Now that high nitrogen content actually persists for a very long time. My colleague, the former colleague, he's not here anymore, Dave Carter, who was a soil biochemist, microbiologist, Dave was able to find different parts of where different parts of a body had been in a field where there'd been a suicide in canids or something had scattered the body parts. And so by taking soil samples and looking at nitrogen content, he could retrace where the different body parts were. According to Dave, there's no question that you can see increases in nitrogen in soil from a large size animal for at least 10 years and possibly longer than that. So it lasts considerable time. I personally don't think there's a lot of difference between active decay and advanced decay. Essentially, it's just a question of how much of the body's gone. But in the advanced decay, most of the soft tissues have been removed. And the question you should ask me is, how long does this take? And the answer I have to give, it depends. It highly depends on temperature, as we're about to see. After advanced decay, we get to the remain stage. And the remain stage is ligaments, tendons, skin, and bones. Eventually, those tendons, skin, and bones will be decomposed. You'll have bones, and then rather than biotic processes being important, it tends to be abiotic processes in weathering. So I mentioned these agents of decomposition. This poor dog got hit by a car on a road in Brazil that we came upon not long after it was hit. And there were vultures feeding on the body, as you can see. And they will take a tremendous amount of tissue. This is the same carcass the day later. So ordinarily that would take many, many days, even in the tropics. But when you have vertebrates consuming a large amount of tissue, you get very rapid soft tissue loss. Among insects, the most important group by far are flies. And of those flies, those in the upper left and center, the blowflies, are the most important. They're the most important because they're the ones that come to the body first. So these are insects in the family Chrysomelidae. There's some others. Your common house flies, they will appear at bodies sometimes. So will what are called flesh flies. I personally dislike flesh flies because, one, I think as a fly, I don't think they're very attractive. So that's an aesthetic judgment. And secondly, about the only way you can identify them is by getting males and dissecting their genitalia. And it's hard enough counting hairs on flies to do species IDs. I don't need to be dealing with male genitalia to ruin my life. So flies are an important group. The other big group are beetles. And these guys with the bulby antennae are carrion beetles. They'll come and feed, and they'll feed at the same time there are maggots there. Some of the others are feed on dung from the cadaver, but the one I really want to make mention of in this slide is in the lower right. Those are dermestid beetles, and some of you, if you've ever been interested in taxidermy and so forth, dermestid colonies are used to clean bones. They'll completely consume all of the soft, dried tissue on bones and leave the bones essentially cleaned. I can virtually guarantee that you guys are living with dermestid beetles in your house. I'd be very surprised if you are living someplace that's so sealed up they can't get in. And mostly what they're feeding on are dead insects. So they're kind of the bane of insect collectors. All right, I mentioned that I couldn't tell you off the top of my head how long it takes to get to a certain stage. We did an experiment with exclusion cages. So here, 2 dead pigs are killed within minutes of each other, set out. The cords, of course, are thermocouples. I didn't mention that before, but we were recording temperatures. Here they are 7 days later. The one on the left is when we allowed access to insects through the cage, and the one on the right, which is in full bloat stage, is one where the insects were excluded. So they make a huge difference in terms of how fast the decomposition process occurs. Well, okay, let's talk about the homicide, and then we'll talk about some of the research that we've done, in relation to it. Not so much in relationship to this case, although there was a scientific paper that we produced that directly was a consequence of our work on this case. But I thought it would be useful to just show the process of how you approach a crime scene as a forensic entomologist, and then show you problematic points in terms of doing the analysis. And that's where the research kicks in. So the decedent was a truck driver who had disappeared. When we found the body, the police had the suspicion it was the missing truck driver who had been gone for more than a week. Unbeknownst to me, they had found his truck at a truck stop in West Lincoln, abandoned with a knife and a blood-filled cap. So they were reasonably certain the truck driver was dead. I didn't know any of that. And in fact, it's a bit problematic trying to get the police not to tell you details about the case when you're working it. So I received a call. Let me go back. So let's see. On the 13th of September, so not too far removed from where we are today, that a body had been found in Seward County by a man walking his dog. So we got the call from the police. And so myself, Fred Baxendale in entomology, and our grad student, Tim Huntington, went to the scene. And as hard as it is for me to believe this happened, when we got there, they had finished taking their photographs and so forth. And essentially, they turned the scene over to us to do the insect collection and insect work for the next 45 minutes to an hour. So when you start, you sort of spiral your way into the scene. You don't want to disturb things. You don't run straight to the body. You can't really see the body, but you see that in the middle of this slides, there are some branches in a ditch, and that is where we're going to find the body. There were lots of blowflies on the plants around the corpse. You can't really use the identification of those flies. You really have to only use the identification of things that you recovered directly from the corpse, but they're still informative. What species of blowfly really matters? I collaborate a lot with Neil Haskell, who's kind of has been the premier forensic entomologist in the US. And one of the things I'm doing with Neil is digitizing his case files. So I've managed to digitize about 450 of 1,600 cases. So here are 18 years worth of cases. And what's noteworthy, the species that he used in almost a quarter of his cases was the same, Phormia regina. Phormia regina actually occurred on this case as well, but it turns out it's a different species that was more informative to us. There are also these blister beetles. They're kind of cool beetles. You can see them on goldenrod. They also occur in alfalfa and various flowering crops. They're called blister beetles because they reflex bleed, and their hemolymph, or blood, actually causes blisters. And so this is one of those instances where we collected them, took pictures. In the case that they had a suspect with unexplained blisters on their arm, we might be able to associate them with the crime scene. So here's the body. Oh, and I should mention, I've been doing this too long, and I'm not sure I was ever that normal to begin with. But I am not at all bothered by working with bodies. I'm a little put off by the smell, but frankly, I had to smell liquefied rabbit. And when a rabbit's decomposed down to the liquid state, it's really, really stinks. It's still not the worst thing I ever smelled. The worst thing I ever smelled was year-old corn that had been allowed to get wet in a trash can. And when we took the lid off, I felt like I was a cartoon character who got punched in the face because that stuff smells terrible. Incidentally, if you're at a crime scene, it's always good to be the person who goes to the body and does collections and then says, okay, you guys can move the body, because the underside of the body, no matter how bad the body smells, the underside will be much, much worse, because that's where the anaerobic microbes are operating. Okay, this slide tells us a lot about the homicide. You can see that there's a standard pattern of decomposition. The head is skeletonized. There's black there from decompositional fluids. It doesn't look like the body's in bloat stage. And there's some curious things. There's a mass of maggots. I don't know if you see my pointer. Yeah. There's a mass of maggots here that really have no business being in the torso. So that's suggestive of a wound of some type. We'll see more about that in a minute. And then as we get close, we can see that there's large maggot mass formation. And this is looking more like a knife wound and less like a gunshot wound. You can see that the skin has slipped off the head or has been eaten away. The jaw is disarticulated. It's really uncommon to collect all the teeth and all the finger bones if you get to advanced decomposition. The jaw is only held by ligaments, and so it always disarticulates as the decomposition goes. And then you can see maggots have moved into the collarbone area and so forth. Let's talk a little bit about this maggot mass, because one of the things that the police wanted to know, and really the number one reason I was there, is to give them some estimate about when the individual was killed. If you ever have the chance to see a maggot mass, I'd recommend using a glove. But take your gloved hand and put it in the mass, and you'll be astonished at how hot it is. Even though insects are cold-blooded, when the larvae gather in a mass, they can generate a lot of metabolic heat. And so it's not uncommon for them to be 10 C or 12 C higher than the ambient temperature. in fact, they can reach temperatures high enough that they're close to the lethal temperature for the insects. These guys are a different species you haven't seen. These are actually maggots that were on that dog I showed you earlier. This is a species, excuse me, this is a species called Chrysomya rufifaces. And rufifaces is a tropical species that's become invasive into the US. And so it occurs now as far north as Michigan, which kind of astonishes me. I had a student, Amber McInnes, and I had said to Amber that I didn't think it was true that maggots, when they're in these masses, were growing at an accelerated rate. It seemed to me that they should be experiencing competition. So we looked at interspecific competition. Here we looked at Lucilia sericata and Phormia regina, and we basically saw that although both species were co-occurring, we didn't see any evidence of competition. If you've not read these replacement series diagrams, where these are just different proportions of the two species, and the closer this intersect line is to one, the more indication is that they're not competitive. In contrast, you can see that these two lines cross more significantly to the left, showing that rufifaces could actually outcompete Lucilia sericata. I didn't put the chart in for the other comparison because the other comparison was interesting in that, irrespective of ratio, when rufifaces was grown with Cochleomya macellaria, the other species we looked at, it killed all of them. So that was sort of the most extreme form of interspecific competition, where it's not just that they're competing for resources, it's that they actually will kill their competitor. It might sound like a trivial question, but I was a little interested about why the maggots were arriving and moving around in maggot masses, because if they're under selective pressure to try to complete their development as quickly as possible, surely it doesn't make sense for them to be moving around. So one hypothesis was that they were actually drowning in their own juices. So I had a student, Melissa Lane, who looked at this question about hypoxia survival, and she did some nice lethal temperature 50 studies. And the thing to note is that this time scale is in hours. So the lethal temperature 50 took hours, even at high temperatures where the metabolic rate is high. So it almost certainly is not hypoxia that's the reason that they whittle. And in fact, I can answer that question by, I didn't show the data, but we looked at tolerance to temperature in various maggot species, and they actually will die at about the same temperatures as the center of a maggot mass will achieve. So almost certainly they're moving to thermoregulate. Another student who recently finished, Fabrice Benossi, answered the question that I've been curious about, whether or not we could produce reasonable maggot masses in the lab and if we could see differences in development. Nobody likes having, I think this is fair because it was true in entomology as well as here. Nobody likes having somebody rear dead animals in their building. And we didn't use animals, we used beef liver as our feeding substrate. But yeah, there's a certain amount of odor that's associated with that, even if you manage your colonies properly. And so, so we're curious about this. And the take-home message from this slide is that there's much faster development at these rates where there aren't, at these densities where there are not large numbers of maggots versus where we have maggots feeding together. And that also was true on survivorship. So it clearly shows that there's intraspecific competition occurring in those masses, and that, along with the temperature differences, even though the temperature is higher, it's not enough to offset the fact that they're actually developing more slowly. So that was an important finding that's pertinent for trying to figure out the answer to this question of how long has the body been there. I thought I would show you two slides to illustrate manner of death. And this also illustrates my mistakes because it never occurred to me to put a scale in these photographs, which I should have done. But you can see that this is now looking at the other side of the body. There's another, what appears to be knife wound. And actually, as we waited and maggots moved around, you could actually see very definitely the pattern for the knife wound. Now, had we not come, maggots essentially would have eaten that area out eventually and destroyed the evidence. So we were fortunate in that. This is Tim Huntington and Larry Barksdale, who was our contact with the police. Tim's recording locations of the different maggots on the body and where we're going to sample. We sampled those maggots, brought them back to the lab. We had to make certain that we maintained chain of custody and all the sorts of things that you see on TV about being able to go into court and testify that nobody could have interfered with the maggots. And that brings me to what has been the real focus of a lot of my research, and that is issues associated with development time of maggots and how we determine those. And I recopied this slide from a talk I gave, 22 years ago, where I griped about how bad the data were that forensic entomologists were using in their cases. And I argued for certain things about how we should approach these questions experimentally. And as part of that talk, I raised this issue of there are all these different sources of variation. And as I look back on this now, my research group actually examined a fair number of those. And we have answers for a lot of the issues that we had identified as problematic in the past. Oh, God help us. Okay, this is where if you're not already asleep, you may want to take a nap. It's necessary to explain what we did for the development studies. It's necessary for me to explain how we modeled development. And it's not very sophisticated modeling, let me tell you. This chart shows a development graph where rate of development is defined as 1 over the number of days in a stage. I should mention that these flies have the egg stage, 3 larval stages, pupal stage, and then the adult. And in the 3rd larval stage, half of it they feed, and the other half they just wander around. They're called migratory thirds. So in terms of our analysis, we treat that migratory 3rd stage as if it were a 4th stage. You'll notice that, maybe you'll notice, that this sort of S-shaped curve, there's a long thing that you could actually approximate with a line. And that's what's actually done in most studies. So the models that are used for development are really valid in these regions for these temperatures where you have this linear development. And so we can approximate those, and there's this long-standing procedure called the x-intercept method, where you can determine what the base temperature you use for calculations, how many degree days. And then that brings open the question of what is a degree day then? Well, because insects are cold-blooded, you can't talk about development in terms of number of days because it's entirely dependent on temperature. So what you do instead is use a combination of time and temperature. Let's say 100 degree days for some developmental event. Then each day, we look at how many degrees there are above some minimum, and that area, if you will, represents the number of degree days. I'm pausing for you to wake up or be ready for the next thing. To show that I wasn't just an arrogant jerk, we took some papers from the literature and looked at their models and their reports, and what we discovered was, while if their degree day models were valid, it shouldn't matter what temperature you measure development. It should be the same number of degree days at 20 as it is at 25. And what we found was that's not the case. We found all of we found studies that had essentially a non-zero slope when we compared temperature versus degree days. So we knew that they were inaccurate. The question becomes then, what is the source of the inaccuracy? One thing we looked at was light, because a lot of these studies were done under 0 constant light conditions. And sure enough, Mike Fisher, working with me, and Paul Nabity both showed with multiple species that there's an interaction between temperature and development based upon light environment. So we went to a 16:8 cycle for our studies, and that, I think, has sort of emerged as the standard. Why people were ever doing studies under constant light, I can't find an answer for that, other than maybe their chambers didn't have the ability for them to regulate the light. The big contribution that I think we made, which is really more Amanda than me, is an amazing study on the development of 2 species, Lucilia sericata and Phormia regina. Amanda and I, when we should have been listening to papers at a net meeting, we actually holed up in I think it was in my hotel room, and wrote this NIJ grant proposal, which we got funded. And it allowed us to do allowed Amanda to do, let's be honest, a really heroic scale study on development. She looked at multiple times of development for each of the life stages of these insects. And to give you just some idea of how large the experiment was, just to keep the colonies and conduct the experiments, we went through 1/4 ton of beef liver to feed the flies. So we ended up dealing with tens of thousands of points of data for development. For comparison, in the literature, most of the studies had maybe 30 or 40 points, if you were lucky. So one of the things that we were immediately able to deal with was the question of stage transitions. There'd been a lot of writing that when maggots go from one stage to the next stage, there's a large push of maggots, then a long tail, sort of a negative binomial distribution, if you know what that means. In fact, we found first that they didn't develop below 7.5. This is for Lucilia sericata. And instead of being skewed to one side, the transitions were normally distributed. That remains a really important finding, because there's lots of the literature that talks about collecting large or oldest insects, which really biases your samples in the wrong way. It's not just adds variability, but it adds bias. So we were able to do these cool stage transition graphs where we could look at what proportion of a certain sample would be with each stage. So presumably you could use that, to determine where you were on the development graph. I also did probit analysis because I wanted to be able to determine the 50% point. So I just used, it's like an LD50 analysis, where I could determine where the midpoint was when the insects were going from one stage to the next. And I had really great fits on those. We did do curvilinear modeling, but the problem with the curvilinear modeling is there's no single model that fits stuff. This is a very strange thing to say, but mechanistically, there's no clear understanding about the relationship of temperature to growth. Growth is increase in cell number, increased cell size, and cell differentiation. And you would think that that's a biochemical process, so presumably the reaction kinetics of those equations would control what's going on. And people have tried to build models based on reaction kinetics, and when tested, they failed. I suspect it's because there's more to it than that. There's also issues about membranes changing their permeability with temperature. And so it's an intrinsically complex problem. What I can say is that with our own comparison of curvilinear modeling versus linear modeling, we did just as well or better with the linear models, which is kind of a relief because it makes the analysis for casework much, much easier. So we developed our lines, like I showed you before, for degree days for Serracata, and we did the same thing for Regina. And we held ourselves to the same standard. If we're going to criticize other people, do our models work? And I'm happy to say that, yeah, our models showed no change in slope. So what does this tell us about our victim? Well, we need to know which species are there. We need to measure exactly what stage they're in, and by taking them back, we would account for the time that they would grow to the next stage, so we could get a pretty good estimate of what their age was. Then we need to go back in time and figure out what were the daily temperatures, and can we calculate then back to see what is going on. Now, having said all that, I knew by looking at this body roughly what the time of death was. And I didn't know it because of the insects. I knew it because it takes a certain amount of time to remove the tissue off the head. And I knew we'd had moderate temperatures in the 60s and 70s, highs in the 60s and 70s. There's, if you look here on the legs, you can see how the legs have gone, the skin's sort of translucent and the legs have gone yellow. That yellow coloration is adipocere. If you're fat like me, when I die and decompose, all that fat gets hydrolyzed and it changes to this waxy, horribly smelling stuff called adipocere. And so the formation of adipocere under most circumstances doesn't start for about a week. So I had it in my head that we were looking at a body that was at least a week old, possibly older than that. I couldn't testify to that, but I could use that as a guideline for my answer. The other thing that was noteworthy is that the body showed signs of skin slippage. This is where the epidermis separates from the dermis, which is a normal part of decomposition. And it sort of is, typically occurs faster than adipocere formation, but it's still in that sequence of events of changes in the body. So we took our temperatures. Tim did some sampling at autopsy to make sure we hadn't missed anything. At autopsy, there was a huge mass of maggots underneath the body, which is what they're showing you there. And you notice how you can see the ribs? That's not the way the body went into the morgue. They actually continued feeding while the body was in the morgue. I don't know if you're aware, but morgue temperatures are something like 36.5 Fahrenheit. So it's not cold enough to stop those maggot masses from feeding. We'd actually put a recording thermocouple in the body bag, and we used the information from that and then some studies we were able to do over at Vet Science with pigs in their coolers to try to estimate how long. We came up with an equation that characterized maggot development in morgue storage if you didn't collect insects at the scene. So that was a positive outcome. We had to put a thermocouple in the field and record temperatures at the actual site. And then we compared those temperatures to the nearest weather station, which in this case was 5 miles away. And I was happy to see that I could do that comparison with a simple linear regression, and there hadn't been any frontal movement in that time, so I didn't have any big disparities in temperatures to deal with. Okay, so we have reared them, we know that time, we know the recovery to rearing, we have the information for the crime scene, we have these different factors to consider. What's the bottom line? I put the parameters in my spreadsheet to do my calculations, and these are the results, that there's 2 blowfly species identified, Cochlea biomasillaria, forma regina. Both of them are right on the verge of migrating. So we knew that they were at the end of that 3rd stage. We were able to correlate our site temperatures with a nearby weather station if 5 miles is nearby. And then we looked for other insects that were consistent with the condition of the body to make certain that there was no evidence of the body having been moved or something like that. And what I came up with was 8.5 days, which was pleasing because Tim did his analysis separately from me and came to the same conclusion. So this is, if you want to read it, this is actually the verbatim conclusion from my case report. I mention it because this is another place where I really screwed up. I put the 8.5 days in there, but what I didn't put in was any indication of variability. And that's kind of a boneheaded mistake I would never do now. But at the time, I'm going to just use the excuse that it was an early case for me, and I wasn't thinking straight. The police had actually had 2 suspects. They had caught the suspects trying to use credit cards from the driver, and so they actually had people in custody before the body had been found. And I'm trying to think. It was a plea of no there was a plea of no contest. I remember being in my office, all ready to go to the courthouse to testify when I got the phone call that the suspect had pled out. The police had been really happy with our results because they had 2 other lines of evidence on time of death. So we had, I didn't mention it here, but one thing that's important about blowflies is they won't lay eggs at night. So we had said, well, it's 8.5 days, which put it at the morning of September 5th, but the death could have occurred during the evening hours. We just knew that it wouldn't have been any earlier than sunrise that morning. Well, it turns out that they had a black box in the tractor, so they knew how far it had been driven different times. And so that mileage corresponded to where the body was. The other thing is that Morant pled out to accessory to second-degree murder, and she said that he had been killed at 2 in the morning. So they had 3 independent lines of evidence showing the same time of death. I guess I should mention, I put this in here. It's not really part of my job, and it's one of the things I've warned my students about, that our job is just to say what the scientific data are and leave it at that and let the court decide. Obviously, we'll say what the implications are, but it's wrong for us to think that we belong to the prosecution or the defense. We're really working for the court if we're working for anybody. Before I finish, Amanda and I have an LLC for doing our consulting work, and so if you want to learn more about this stuff, you can go to deathanddecomp.com and go to the resource page. I am very proud of this write-up I did about why aliens did not mutilate cattle in Colorado. So that's worth a read. But thanks so much for your attention. I'd be happy to answer any questions.
Any questions? I'll bring that mic around and carry on.
Thank you for the talk. This is fascinating. I'm just wondering, since you mentioned there's a knife wound in the victim's body, so why didn't the magnets get to that part? Because the flesh was exposed. So why didn't they feed on that? But
they had actually been feeding there. That one slide I showed you where you could see the shape of the blade, they had been in there. And as we moved the body and removed the covering, it exposed it to the light, and they were kind of, I don't know whether I say bashful, but they moved away and moved out of that site and moved around. When the body went into the body bag, all of the maggots that had been at different places on the body essentially went right underneath, moving to form one giant mass. And so if they were actually in there, they just had moved out.
But you can still tell the kind of wound, the shape and things, even without the feeding.
Yes, you can. Yes, you can. That wasn't true for all the wounds, but
I
it was
see.
true for that wound.
Thanks. And also, I wonder if there are other ways to complement this method of judging the time by the insects. Is, for instance, because you mentioned like bacteria early on, is there like methods using bacteria, or is most useful to use insects?
It's most useful to use insects. The main problem with insects are that we can't precisely estimate their time in a stage. There's people who have been working on cuticular hydrocarbons, and they've been trying to show that the cuticular hydrocarbons vary through time. And so you could do a GCMS test on, basically you're testing lipids on the outside of the insect. And if it's accurate, that might tell you that they're at the beginning or the middle of a stage rather than someplace in between. A lot of the variability in an estimate comes from not knowing exactly where in the stage the insects are. So that's one technique that's used. People have looked at molecular markers, but they've not come up with anything yet that's reliable as a molecular clock. So.
Thank you.
You're welcome.
I wanted to ask you a little bit more about like how the flies find dead bodies. But when I was in Namibia for the year, I was out with students and we had to dig a trench to do our business in. And so I did my own little experiment because they've got these dung beetles that are pretty big and fly. And so it took 3 minutes from the time I finished my business to the time the first little hummingbird sized thing flew, which I thought was just pretty impressive. I mean, maybe they're, anyway, and I know that's a different thing, but
It's the same idea because it's essentially exactly the same idea that feces is a limited resource. Interestingly, there was an evolutionary question about which came first, searching for poop or searching for bodies. And I think the consensus right now among entomologists who study that is that it appears that it was searching for the species that search for bodies evolutionarily preceded those that search for feces. But I remember being in Brazil after a monkey troop went by and they had defecated. And oh my God, there were so many insects on those little pellets of dung, each fighting to try to get theirs. And so it's the same thing, transient resource that they want to get. But boy, I envy you getting to see those big beetles.
I just have a kind of naive question. Will those insects attract some animals eat them?
Oh, that will eat them?
Yeah, like
Yeah,
animals will
they
come to
do.
eat them, and will that affect?
Yeah, mostly it's other insects, but there are beetles that will come to dead bodies specifically to feed on maggots. And I don't want to ruin anybody's perceptions, but butterflies often come to dead bodies and feed on the decompositional fluids. Butterflies have trouble with regulating their salt balance, and so they apparently are deficient. This is a generic issue with butterflies. And butterfly collectors know this because they'll often pee on the ground where they think there are butterflies and wait for butterflies to come and start feeding on the urine pool. And so you'll also sometimes see clusters of butterflies on a salt flat or something. So, but I've seen beetles come and attack. I've seen wasps come and attack maggots. There's an argument that the mass is partially defensive, that there's so many there that it discourages predation. That seems kind of counterintuitive to me. I would think that because there's so many, you would see predators there. But for the most part, you don't see, you see a few predators, but not nearly as many as you would think there would be relative to the size of the resource.
Thank you.
Yeah.
I got one more. It just occurred to me, Rand made me think of, so the life stage of these larval flies is there a chance that they run out of dead body before they, and run out of food before they have a chance to become a fly?
Yeah, and in fact, there's, I didn't show it, but my student, Tim Huntington, Tim did a study. He was interested if you could get a second group of flies laying eggs, because it always seemed like, we were always curious about what the maggots we saw, how could we be certain they were the first ones there? Maybe there would be a second or third one. And they have a strong, once the maggots are established in feeding, there's a strong antithesis, or what do I want to word? They simply will not lay eggs on the same body. And I assume it's because of that competition issue, Larkin. So for the ones that are there, especially with a large corpse, they have enough. There's definitely a lot of mortality, which is a good thing, or the